Hello, my name is Hanif. I'm from Institute of Planetary Survival for Sustainable Wellbeing in IIUM. So I'm presenting my, a whole team project and addressing the challenge of Candida auris in a changing climate with a DNA biomarker-based diagnostic approach. So the aims here is actually together to bringing the gap between the clinician and the scientist, which I'm from, where we are looking into to simplify the diagnostic step, where in conventional method, one at a time, at this proof of concept or technology that I'm integrating here can detecting more than one at a time for specifically Candida auris. Next. Okay, so Candida auris is a pathogenic yeast that has gained attention due to its emergence as a significant healthcare-associated infections. So the fungus threatens vulnerable population, patients, particularly in healthcare setting, cancer patients, immunocompromised patients, and it's mainly resistant to multiple antifungal drugs. That means the drug's not working to treat these climate-induced species. So the emergence of Candida auris has been linked to climate change, specifically global warming, which has to be suggested to act as a selective pressure on these pathogens. Next slide, please. So the hypothesis here is actually the climate change had been induced Candida auris adaptability to thermal and then transform it to become a pathogenic species. So this adaptability combined with the ability to survive and then to spread in warmer environments, perhaps by birds across the globe to rural areas, and then where people and birds are in the constant context. And what else? When we look at the diagram here, or the picture here, rural environments where we mainly found that farming activity provide all the opportunity for interspecies transmission of virulent pathogenic species of Candida auris. And then the next carry on with urban migration. People move to the city carrying some of these pathogenic species. Human migrations towards urban area eventually leads Candida auris into healthcare environments. So in the next slide, I will show you what's happening in the healthcare setting. So a study found that delayed treatment for candidemia, that means the candida that been infecting the blood, causing the septic shock, was an independent predictor to mortality. If you look at the diagram here, the relationship between time to treatment for invasive candidiasis and hospital mortality increased more or less around 20% for any delay of 12 hours treatments, effective treatment to this patient. So this highlights the importance of timely antifungal treatment in management of candidemia. Next, please. So in response to this complex challenge, our research explored the potential of biomarker detection platform as a simple nucleic acid biomarkers or biosensor for identifying Candida species, including specific identification of Candida auris. Please. So this is a DNA blueprints of Candida auris that we try to target, that we can make it one at a time. We change one at a time to two at a time for detection or quicker detection of Candida auris in clinical setting, that we help clinicians identify the Candida auris and then provide appropriate treatment for this for the patients. Okay. So our approach-based duplex maybe sounds a bit, lab-based, but eventually what I want to show here is basically we can detect many targets, many species in a single tube in one reaction that tremendously will affect the outcome for the patient. Okay, so the graph here showing the duplex or double detection at a single analysis of a patient result. As you can see here, the species specific is, we can replace it to whatever species I want. But in this particular research, I'm focusing on Candida auris that can be, manipulating that peak at 26 degrees Celsius. And whereas another one is for pan-Candida probe that can covering a broad range of pathogenic Candida species at 45 degrees Celsius. I will show you some result to what happened when we detect Candida auris in laboratory. So what's happened here, whenever that you detect Candida auris, the peak or the markers at 26 degrees Celsius, it drops significantly to show that these species have been detected in the tube or in the reaction. Okay, so, and the 45 degrees Celsius showing that there is a genus detection of Candida auris in general. Okay, in conclusion, this is a preliminary result that we are looking forward for more collaborations to meet the clinician that we can test the assay further in a real patient sample. Next, please. Okay, so this is some people behind the project. Next slide, please. And special thanks to Professor Mohamad that provide all the facilities and the amenities for my research, and the rest of the team from Liverpool School of Tropical Medicine and the grant. And thank you very much for all of you.